This is a huge moment in science and also in medicine because CRISPR itself, this gene editing tool, was really only discovered almost a decade ago. It won the Nobel Prize in 2020, and sickle cell itself has also been a neglected disease in the pharmaceutical industry. So this is a huge moment on multiple fronts. Check it out.
2 years ago, it was hard to imagine 15-year-old Johnny Lubin doing this for very long. Born with sickle cell disease, an inherited disorder affecting the red blood cells, Johnny has been in and out of the hospital his entire life, dealing with bouts of extreme pain and other serious complications.
It was kind of hard for me to do things like have fun and stuff because I'd always have to be worried about if I'd have a pain crisis or not.
Until now, the only hope for a cure for the estimated 100,000 people in the U.S. with the disease has been a bone marrow transplant. But like more than 80% of patients with sickle cell, Johnny couldn't find a donor.
We were desperate. At that point, we were like, okay, what's gonna be next? We thought that we were gonna lose them.
So Johnny and his family decided to try something that's almost never been done before.
I was worrying that I might be get superpowers or
As part of a clinical trial for a completely new kind of treatment, Johnny is now one of the first people in the world to have his genes edited using CRISPR to treat his disease.
And I'm like, wow, that's pretty cool.
And scary.
And freaky.
Do you feel like a medical pioneer?
I don't know. I feel like a guinea pig.
In 1951, Henrietta Lacks came to Johns Hopkins Hospital with cervical cancer. While there, doctors removed some of her tumor cells and discovered they could thrive in a lab. It was something human cells had never done before.
This biopsy taken from Henrietta Lacks produced a cell line that would grow essentially forever and was immediately distributed all over the world.
Named HeLa cells after Lacks, they were used by Jonas Salk to discover the polio vaccine. They also aided research in cancer, AIDS, and diabetes. Dr. Francis Collins, director of the National Institutes of Health, says they've saved millions of lives.
This is everywhere.
But Lacks, poor and uneducated, died in the hospital, and the scientists took the cells without her consent.
I said, my grandmother is not just a cell, she's a person.
And now, 62 years later, the National Institutes of Health is giving them a say. Going forward, all research of the Lacks genome will be approved by a panel with 2 Lacks family members on it.
In sickle cell, a genetic mutation causes red blood cells, which carry oxygen around the body, to be misshapen, like crescents or sickles. They can get stuck in the blood vessels, causing severe pain and decreased oxygen to organs. CRISPR allows you to make a precise cut in DNA. In this case, cells are removed from the body and edited to turn on production of a different form of the oxygen-carrying protein hemoglobin, a type we have when we're babies, explains Johnny's doctor, Monica Bhatia. Then the edited cells are returned to the patient.
So in essence, it's a fetal hemoglobin induction process.
And that's good enough?
Fetal hemoglobin, we know, has oxygen, a higher oxygen carrying capacity than adult hemoglobin or sickle hemoglobin. And so yes, it is more than good enough.
And so far, it has been good enough. 29 out of 30 patients, including Johnny, met the trial's goal, being free from having a pain crisis for at least a year after treatment. How long has it been?
2 years. 2 years.
Now Johnny and his family celebrate his treatment day as his second birthday.
And October 4th is when I got the infusion. So basically I got the whole new dose of cells and stuff.
You teach them how to drive, so that's another things to worry about. So yeah, so we're stepping into the regular worrisome of raising a teenager.